Yeah, there was a problem in the electricity plug point. Okay. So in this, the leaf structures will be given, and then according to the leaf structures, the adaptations are taken place of all these three pathways. Now I will share the slides. Okay, are you seeing this one?
Yes.
Yeah. So this is photosynthesis. So the major activities or major inputs are four: carbon dioxide, water, light, and green pigments in chloroplast. In some plants, even without these green pigments, photosynthesis takes place. I will let you know how it happens at the end of my class. Okay? That output of the photosynthesis are two major components, carbohydrates and oxygen. But the water also as an output it will go. Okay? So in this within this only we are today I'm going to teach. It is water is taken by the roots. Okay, then it has been transferred to the plants. I mean leaves. So this is the particular leaf structure we have to study in deep. I will teach you later on. So within this leaf, the chloroplast major role. A major role. Then with the water and the atmospheric carbon dioxide, this become results of photosynthesis. Then process, how it takes place. This is actually an oxidation process. Initially, water is oxidized, CO it is reduced. Okay, then the oxidation and reduction process, two molecules are byproduct. But the reduction actually aiming for fixing of carbon dioxide with the molecule, and then it makes to make it to food. So initially the carbohydrate reduction needs assimilation of powers. So it needs some power, especially ATP and NDP. So the reduction of carbon dioxide occurs in dark reaction. That means it is not dependent on light. It doesn't mean that it is taking place only in the dark. But generally we call that is dark reaction. Okay, it is light independent reaction. Then that's what we say light independent reaction. To use assimilatory powers of ATP and NADPH, we need light. Okay, so this is light dependent. There are two activities. One is light dependent. Light independent and light dependent. So when it is light dependent, it should be all day. Light dependent, it can be taking place with a day or night. So this is two processes seen. See, light dependent phase, light independent. Light dependent phase, the light reaction or Hill reaction. Okay. Then light reaction or Hill reaction. Light independent phase is dark reaction. or black mass reaction. So we wanted to study these all reactions one by one. So there are three different pathways in the there are three different pathways in the okay. There are three major reactions. One is Kelvin reaction. The other one is Hutch and Slack cycle. The other one is Chem cycle. So all these three cycles are taken place in different plants. The Kelvin cycle, the end productive is C3, 3-carbon compound. The Head and Slack cycle, that is C4 cycle, that is end product is C4 plant. Then Chem cycle is crystallization acid mechanism. So this is also a carbon derivative. So all these three cycles I want to teach you today. Now, the dark reaction, I have already told you, the dark reaction is Kelvin reaction. See, so the Kelvin cycle can be called in all these names. See, Kelvin cycle, Basem and Kelvin cycle, Blackman reaction, carbon assimilation, path of carbon in photosynthesis, reductive pentose phosphate cycle. So all these names implies the dark reaction. That means it is light independent. Okay, light independent. But keep in mind, to have this reaction, we need energy, ATP and NADPH, that has to come through the light reaction. So once the light reaction is there only, it can proceed further. Understood? Understood? Please.
Yes.
Yeah. So there are three major cycles: C3 plants, C4 plants, and Chem plants. But C3 plants is light independent. That is generally called dark reaction. Kelvin cycle, Bestman and Kelvin cycle, Blackman's reaction, carbon assimilation, a carbon photosynthesis, a reductive pentose phosphate cycle, all needs energy which is produced from the light reaction. Okay, so Kelvin cycle consists of two important steps. One, careful, listen. One, synthesis of carbohydrate, then regeneration of RUDP. These two taking place in 3 steps. Major 2 steps, but these two will be taking place in 3 steps. So this is all really enzymatic reactions. So I will go one by one, but you have to memorize later by writing on your notes and study each step is very clear, because if I started to explain all these things, the lecture time may not be available. Okay, so this slide you later on you read, but after this I will comprehend in 3 major steps. Okay, the synthesis of carbohydrate. The second one is regeneration of fibulose phosphate. See, this synthesis of carbohydrate and then regeneration of RUDP. Okay, this is first and this is second. I will explain now. See, 3 phases of Kelvin cycle. I told you there are 2 steps, but phases. See, first one is constellative phase. The second one is reductive phase. The third one is regenerative phase or regeneration of fibulose diphosphate. See, in this cycle, Stage 1, stage 2, stage 3. These all are under 2 steps. One is carbohydrate synthesis and regeneration of glucose. Now we will see. So now the 3C molecules are coming. And in the stage 1, it takes 6 ATP molecules for the synthesis. This is what I told you, this for the 6 ATP molecules are produced by the light reaction. It comes, then second phase, reduction phase, where the NADPH is reduced to NADP plus. Okay, then it enters into the third step, regeneration of RUDP. Okay, there also we need energy. Then it completes. So this is a simplest form of Kelvin cycle, but there are very elaborate steps in the Kelvin cycle. You have to get down that now, very elaborated steps, but you have to see that these two, maybe several steps. These steps are in big carbon cycle. I don't want to explain that. You can go to study for that. Okay. Now, so in these 3 phases, there are several enzymes. See, in this phase 1, 2, 3, there are several enzymes involved. I have already told you this is purely enzymatic reaction, Kelvin cycle. So we will see what are the enzymes involved in the Kelvin cycle. In this first carboxylative phase, only one enzyme, carboxy dismutase or dismutase. Okay, carboxy dismutase. This particular enzyme catalyzes tribulase 1,5-diphosphate to form PGA, phosphoroglyceric acid. Okay, this is the first enzyme in first phase, only one enzyme. In the second reductive phase, I have already told you, it is a reducing phase. Here, there are 3 enzymes: phosphoglycerol kinase, tribose phosphate dehydrogenase, and then triose phosphate isomerase. So in first phase 1 enzyme, second phase 3 enzyme, and the third phase 7 enzymes. So totally how much? 7, 3 plus 1. 11 enzymes are involved in Kelvin cycle in 3 different phases. Understood? Hello?
Yes, sir.
Yeah. How many enzymes are in first phase? 1. How many enzymes are in second phase?
3.
3. And how many enzymes are in fourth phase? Sorry, third phase? 7. So total how many enzymes?
11.
11 enzymes are needed to complete this Kelvin cycle. Okay? Now, that completes the C3 cycle, Kelvin cycle. Your work is to add a complete reactions of C3 activities or C3 cycle in the notes. Okay? So these all 11 enzymes, there may be 11 steps. So all these steps you have to clearly taken from the textbooks and write down very clearly. And then once you go through slowly only, you will understand all these cycles carefully. We also struggled to study during our period, but later on we have studied, but it's difficult to keep remember, but for the exam point of view, you have to study and keep it remembrance. Okay? Now, I will go for the C4 cycle, that is hat and slug cycle. Why it is called the C4 cycle? The end product, first end product is C4 component. Okay, I will teach you. Okay, dicarboxylic acid cycle. Dicarboxylic means it has 4 carbon molecules. Okay, see, this is up to 1965. There was only believed that only C3 cycles are available in the plants. Melvin Kelvin was awarded Nobel Prize in 1961 in C3 plants, C3 pathways. See, how many years back, maybe around 50 years back, he has clearly indicated the C3 cycle. Then after that, 1965 only, Heath and Slack, they have identified C4 focus. So they have used isomers, that isotopic element, C14 carbon dioxide. Then they have identified 4 carbon compound. That means dicarboxylic acid, malate, and aspartate. Okay, these all are 4 carbon compounds. Okay, then in sugarcane, they found plants are called C4 plants. Okay, because C4 carbon containing dicarboxylic acids are the earliest product. Okay, this is what it contains C4 pathway. That's what they say. These are the C4 plants. Carefully remember: sugarcane, maize, sorghum, pearl millet. You know pearl millet? Have you seen pearl millet?
Yes. Yes.
Can anyone say? Kambu.
Yes.
In Tamil, kambu. Okay. Amaranthus, Panicum mexicum. This is thinai. Chloris, Atriplex, Digitaria. These all plants we have common up to this in Sri Lanka. Okay, we have C4 plants. You see amaranthus. In 21 days you can harvest. Quick photosynthesis assimilation. Okay, all these are, you can similar type: sugarcane, maize, sorghum, pearl millet. You can see the leaves are almost similar except amaranthus. Amaranthus is dicotyledon. All these are monocotyledon. So it is revealed that all dicotyledon and monocotyledon, they have C4 pathway. And these are the families: Cyperaceae, Amaranthaceae, Cynopodiaceae, Compositae, Euphorbiaceae, Cotulaceae. These all families you have C4 plants. Okay, so there are C3 plants and C4 plants. In general, other plants are almost C3 plants because they are bioproductive C3. Here, the byproduct is 4 carbon compound. Now, there should be some structural peculiarities of C4 plants because in general, C3 pathway is taken place, but to have C4 pathway, there are different structural formation is necessary. Okay, these structures, I will see, go to the next slide and coming to this slide to explain the reality of the difference between C3 plants and C4 plants. So the presence of bundle seed cells containing chloroplast. In general, C3 plants, bundle sheath having less chloroplast, but here it has more chloroplast. This is C3 plant, okay, and this is C4 plant leaf cross section. Okay, this is the leaf cross section of C3 plant and C4 plant. So there are difference. In this difference only I am going to teach now. Okay. So the chlorophyll pigment, see, the presence of bundle sheath cells containing chloroplast. Where are the bundle sheaths here? See the bundle sheath. This is bundle sheath. Here the bundle sheath does not have chloroplast. Here the bundle sheath shall have chloroplast. See this picture. Are you visible?
Yes.
Hello? Yeah. This is a generalized picture of particular leaf. This you have to draw carefully. It may be definitely supportive for your essay type of questions or structural type of questions. So this is the part. See, this is, I have already taught you regarding this stomata. Okay? Stomata opening and guard cell I have already given. Then waxy cuticle. This main slow, very thin is waxy cuticle. Then upper epidermis. Normally, we don't have chlorophyll in this portion, but we have chlorophyll in these two. Okay. Generally, in C3 plants, we don't have much chlorophyll in this spongy mesophyll, but in C3 plants, see, all have chlorophyll. Okay, can you see? Green color?
Yes, sir.
The green color indicates the availability of chlorophyll. Okay, so this is the first advantage of the C3 peculiarity. Then bundle sheath cells are radially arranged around the vascular bundle. See, this is radially arranged through the vascular bundle. This is vascular bundle. This is vascular bundle. But in C4 plants, it is radially arranged around the vascular bundle. But here, it is not such that. If it is radially arranged, it is quick transport of photosynthetic assimilation. Oh, the water, everything is very quick. That's what the C4 plants produce more photosynthates in a shorter period because of this particular arrangement. Then, like grana in their chloroplast, if it is grana, see, where is that? I have taught you the chloroplast structure. So this is grana and granum or grana, and then this is stroma. This particular granum is less in the C4 plants. Then, bundle sheath cells remain surrounded by one or more rat-like layers of mesophyll. I have already. Then, sixth one. The mesophyll cells have well-developed granular constitution. This is called dimorphism of chloroplast. The mesophyll cells develop because the mesophyll cells are photosynthetic region. So in this area, in C4 plant will develop and very good pigmentations. Okay, like that you can study the 10 according to that picture and the whole picture. You can go and study all others. We will see in some tutorials. We will teach you very clearly. Then where it occurs, the C4 pathway, chloroplast of mesophyll cells, chloroplast of bundle sheath shell. Chloroplast of mesophyll cell, all these. Okay. See, chlorophyll is bundle sheets. Here only, the photosynthesis takes place in C3 plants. And then bundle sheet cells. The bundle sheet cells, see the bundle sheet cells. Okay, so here only the C3 plants we are having synthetic place. Two bundle sheet cells, mesophyls. Okay, so then what is happening in the mesophyll cells? I have given. Okay, and here you can see this is bundle sheet cells and mesophyll cells. I will explain. Here, the bundle sheet cells generally C3 cycle takes place. In all three reactions, C3 cycle is there. But in addition to that, it takes place, immediately changing the C4 pathway. That is the C4 plant. Okay, so the end product will not be persisting in C3 form in C4 plants. But in C3 plants, it remains as such. Later on, it can be converted into sucrose molecules. But here, in these two cells, take place in this part. Okay, initially C3 takes place, and then it immediately go to mesophyll cells to complete the cycle. Okay. Then reaction, bundle sheet cells also, there are some reactions. Once I teach you, you may not be understanding. You just go through slowly, sit and slowly move and get the understanding. If you don't have any understanding, please contact me. I will, step by step, I will inform you. See, in this picture, it will explain slowly. You read, see this, what is happening, okay? Okay, this is how the cycle is completed. When you go through this slide, once I said you, again you see the carbon dioxide comes, and then another carbon dioxide molecules will accumulate here, and then it takes oxalate acetate, then it comes to aspartase, then malate. Okay, these two are four carbon molecules. Then it comes to the bundle sheet cell. This malate comes. Then another CO2 is added. Then 6 carbon intermediate forms. And then phosphoglycerate is active. Okay, then Calvin cycle completes. Then ribulose phosphate is again, see, this is a cyclic reaction. It takes place. Then carboxylate. Then this pyruvate is formed. Then pyruvate comes. Again, this phenol pyruvate takes place. So this is a completion of C4 cycle. You can read it and memorize it. Okay. Then now we will see what are the physiological significance of C4 plants. It is a must. We will see one by one. Okay. Are you there?
Yes, sir.
Yeah. All six. How many people now? Today now join 125. So good. All how many people the class?
136.
136. One is absent. Okay. So no problem. So now I have seen C3 pathway, Kelvin cycle, and I have seen CO pathway, how it comes. We have diagrams of two structures, C3 plants and C4 plants. There are difference, and where that C4 is taken and how the C4 plants pathway is completed along with the C3 pathway. So now we have come to the how the C4 plants are significant in the plants in a physiological aspect. We'll see one by one. In general, what is that C4 plant? It has more photosynthetic rate. Understood? Hello?
Yes.
Yeah. It is a basic in C3 plants that there are photosynthesis. There are two reactions, light independent and light dependent reaction, dark reaction and light reaction. That is where all the plants they have. Even in the C4 plants, the C3 cycle is completed. Okay, but the C4 plant is very quick to produce photosynthesis. So because of these special structures in the leaf, that the structural formulations make more advantages to produce more photosynthesis from the available light. So these advantages we will see one by one. Okay. Now see adaptation mechanism to xerophytic plants. So once you see this, in the xerophytic plants, very low concentration of CO2. Okay. It can produce very, very little concentration of CO2 also. The CO3 plants, sorry, C4 plants can produce C4 assimilates and aspartate everything. So therefore, the partial closure of stomata in what happened due to xerophytic conditions, the availability of the water is almost less. I told you, the dryness, physiological dryness, even though water is available in some plants, or even without water is available, the xerophytic plants are surviving because of the C4 pathway they have. Okay, even with less, the closure of stomata will not affect the photosynthesis of this xerophyte. Okay, so it can grow lower water content, high temperature, and bright light intensities. It can produce photosynthesis through C4 pathway. Therefore, it is highly advantageous for the xerophytic plants. The other advantage is in the C4 pathway, photorespiration is absent. That means while the photosynthesis is happening, there is no photorespiration. I will teach the photorespiration in the next class. Okay? Then photosynthetic rate remains higher. Once the photosynthetic rate is higher, the xerophyte plants can survive. Then, in general, the C4 plants are about twice as efficient as C3 plants in converting solar energy into carbohydrate or dry matter. This is very, very much important. So C4 plants, okay, C4 plants are able to produce twice the size amount of carbohydrates than the C3 plants. Understand? Hello?
Yes.
Yeah, because only one girl is answering. What about the others? Hello?
Yes, sir. Yes, sir.
Can any boy can answer?
Yes.
You are always answering that. What about the others? They also should participate in my class. Are all visible? That is my worry. Okay. So now I will come. In the presence of the low light, low water content, high temperature, bright light, whatever it is in the xerophytic plants, they have C4 pathway. Therefore, it can produce the efficient photosynthetic. Than the C3 plants, it is comparatively twice the amount of C3 plants. Understood? I will go to the next significance. Okay. Now, the other structure. Crassulation acid metabolism. Chem. Crassulation C acid metabolism. Chem. So with this, I have completed C3, C4. Okay. So C4 in general have a peculiar leaf structure, and they produce C4 compounds at the byproduct, and it's more efficient than C3 plants. Therefore, the xerophytes and the plants can live by this mechanism. General C4 plants, they are producing more photosynthesis than the C3 plants. Example, sugar, maize. We have kambu, millet, and those plants are C3 plants. Now, I am covering the third one, chem plants. Okay? Chem. What do you mean by chem? Crassulation acid metabolism. There's a peculiar metabolism which causes crassulation acid metabolism. See, in natural condition, some halophytic, that means succulent plants, okay, produce acid in the night. Okay? See, the semisucculent plants increases and decreases during daytime this particular acid. This acid is found is this particular diurnal change. What do you mean by diurnal change? Day and night change. Okay? The acidity in green shoots, okay, increases at night and decreases in daytime. It's found in certain family, that is Crassulaceae family. Crassulaceae family. You know Bryophyllum? Tamil in Sadai Harachan, Bryophyllum. This plant is belonging to Crassulaceae family. So this particular acid, crassulation acid, this particular acidity in green shoots, we call crassulation acid. So this mechanism is called crassulation acid metabolism. This is chem. Okay, chem means, so now C3, C4, chem. Okay. See, in the only in green organs in families like Crassulaceae, Cactaceae. You know what is Cactaceae? Cactus? Cactus 0. And Orchidaceae, orchids. And Bromelaceae, Liliaceae, Asclepidaceae, Vitaceae, and Euphorbiaceae. So in these families, this mechanism is prevalent. It doesn't mean that in these families, 3C4 are not available. So, but in these families also they have chem mechanism, because the Crassulaceae family, but all other families also having this particular mechanism, they call CAM, chem. Okay. All such plants are called Crassulaceae. That's what I am saying. In all this family, the plants which have that crustacean acid metabolism. In general, commercial crop, you can see this pineapple. So in general, C3 plants are leguminous plants, okay, normal plants, chili, and then brinjal, and all those are C3. Maize, sugarcane, millets, panicum, not millets, that kambu, pearl millet, and the panicum are C4. And certain families, they have chem. But in commercial, pineapple is an example for chem. Okay? Understood?
Yes, sir.
Good.
Yes, sir.
Yeah, now mechanism of cycle. So this also you have to study. Okay? So this is the ultimate product you can see, and you can study by your own. You can have an understanding. If I explain, that is difficult for you. The leaves of chem plants also contain enzymes of Calvin cycle. I told you, no, only C3, C4, chem. They have C3 cycle. That is a must. In addition to that, they have C4 and chem cycle. Okay? So here we have C. So this is the chem cycle. When I send this slide, you can see this chem cycle. It is not much important. Then chemosynthesis. What do you mean by chemosynthesis? Yeah, what is the time now? I don't have wrote the words. What is the time now?
9:46.
9:46. So then I have to go very quickly. So you have any class after 10? Hello?
Yes, sir.
You have by 10, huh? 1 minute. Okay. So chemosynthesis means chemical synthesis. The energy for the photosynthesis is taken place from the light reaction. I have already told you, no? Light dependent reaction. The assimilatory ATP is synthesized. But in some microorganisms, the light takes place, the energy takes place through the chemical reaction. One, the nitrifying bacteria, the Nitrosomonas and Nitrococcus, these two oxidize ammonia to nitrate and chemical energy is released. See, 2 NH3 molecule and oxidized by 3 oxygen and 2 HNO3 and HNO2 nitrite and water, and the chemical energy is released. This particular energy is used for food production. Sulfur bacteria. Here the sulfur H2S, sulfur dioxide, is hydrogenated and then chemical energy is formed for food synthesis. Then iron bacteria. Here the ferrous ion to ferric ion and release chemical energy. These are two. Ferrobacillus. Here Biggertoia. So these are the bacteria. Okay. Iron bacteria. So these 3 bacteria are producing energy, chemical energy, for the use of production of food. So now factors influencing the rate of photosynthesis. So how many cycles we have seen now, up to now?
3.
3 and 4, chemosynthesis also, no? So the real pathway are 3 we have seen. What are those?
C3, C4 and chem.
Chem. What can anyone answer? What is a C3 plant? Can anyone say? C3 plant? Any plant? Huh? Tell me. Don't worry. Tell. C3 plant. I told you, leguminous plants. Can anyone say one leguminous plant?
Groundnut.
Yeah, groundnut. Why you are saying groundnut? Ayyo, groundnut ke na? Are groundnut. You say groundnut. Say it again. Hello. Say it again. Groundnut. Hello. Where are you all? Okay. Someone else? Can you say groundnut?
Groundnut.
Okay. Be happy in the class. Okay. Now, can anyone say C4 plant?
Maize.
Yeah, maize, sugarcane, pearl millet. Can anyone say commercial camp plant?
Pineapple.
Very good. So understood. Why we call C3? Because of the end product, initial end product is C3 compound. And why do we call the C4 plants? End plant is carbon, 4 carbon compound. Immediate end product, okay? Final end product, all are sucrose and glucose and sucrose and everything. But the initial end product is C4. And then why we call crystallization acid mechanism? The diurnal change, there's some acid formation in the green part is taken place during night, and the day it is reducing. That's what we call, it was found in crystallization family. Therefore, the mechanism is called crystallization acid mechanism. Okay? Understood? Finally, we have seen chemosynthesis. That means the chemical energy are used to food production. Nitrifying bacteria, sulfur bacteria, and iron bacteria. Okay? Now, I will count the factors influencing rate of photosynthesis. There are, as usual, there are internal and external factors. Always there are internal factors and external factors. The internal factor, chlorophyll. So the amount of chlorophyll is directly related with the rate of photosynthesis. If the contain more chlorophyll, photosynthesis more. Example, the C4 plant, chlorophyll, they have more chlorophyll. Okay, that's what it has have more photosynthesis. So definitely the chlorophyll availability will increase photosynthesis. Then enzymatic system. The enzymatic, all you know, the C3, there are 11 enzymes. So then the enzymes are, the amount of enzymes is more, the activities are also more. Then leaf resistance. The meaning of leaf resistance too high, it will open. So if there are gas exchange resist this stomatal opening, that is called leaf resistance. Okay, so that's what we call stomatal resistance. See, stomatal resistance. When the humidity is more and the environment condition more, and there is leaf resistance, that also decreases or increases photosynthesis. And demand for photosynthesis. How many food we need, according to that, we need more photosynthesis. Example, young leaves, young twigs, they need more photosynthesis because to grow. So once you clip the